Excellent, everybody. Okay, well, it looks like we're settling into our seats, and I am very pleased to start us off today with our latest in our webinar series. And we're actually joined by a wonderful team that has been sharing with us some of the work that they've been doing at MIT, the MIT Abdul, Latif, Jameel Water Food Systems Lab, J-WAFS. And so just to get us started here with some housekeeping, this meeting is recorded. So and we will be able to post this to NAWI's YouTube page. So for those of you who miss it, or if you want to go back and listen to some things, it will be available there. We have you muted during the duration of the presentation. However, I often notice that great questions come up as people are presenting. So I encourage you to put your questions and comments in the chat as they come to you. And then as we wrap up the three speakers, we have three speakers today, we will then have a bracket of time and we'll go through some Q&A. And in fact, I may invite you to unmute yourself and ask your questions directly of our presenters today. Also, I just want to remind you that NAWI has an ever-growing body of people on its LinkedIn page. And this is a great resource for you, not the least of which is this is where all of our future webinars are announced. And then as we get publications, we typically point out those publications and announce them on our LinkedIn page. So with that, let me introduce Longyi Han. Longyi is the Director of External Relations at the Abdul Latif Jameel Water and Food Systems Lab. And in that capacity, she works with corporate, government, and nonprofit partners to support and engage in the J-WAFS program. Her bachelor's degree is in Law and Commerce from the University of Melbourne. And then she came to Boston for her master's degree. And so, Longyi, welcome. And I'm going to turn it over to you so you can introduce our three speakers and take us through today's program. Thanks so much for coming.
Great, thank you. Let me share my slide. Great. All right, well, thank you very much for giving us the opportunity to present and engage with the NAWI community again. We are all very pleased to be here. And as you mentioned, I'm the Director of External Relations at J-WAFS. And if you scan the QR code on the slide here, you'll be directed to our website where you can learn more about us. So today I'll start with a brief overview of J-WAFS, and then you will hear from the real stars of today's session, which are our MIT researchers, and I'll let them introduce themselves shortly. So J-WAFS was founded and launched in 2014, and we celebrated, in fact, our 10th year anniversary earlier in the year. We report directly to the Vice President for Research here at MIT, and we work across all the different schools and the 50+ labs on campus. Our role is to catalyze MIT research, innovation, and technology to preserve water and food resources for human need. And this diagram here really helps summarize our impact. And in a nutshell, over the last 10 years, we have awarded $25 million to our MIT community, engaged 400 researchers, and worked with across 40 departments, labs, and centers. And our researchers have also gone on to raise more than $25 million in follow-on funding, and we have supported 12 spin-outs and 2 products. So we do this through 3 main mechanisms. First, we fund cutting-edge research and related activities across MIT, such as seed grants, solution grants, grand challenge grants. We also support students in a lot of their activities through fellowships, prizes, clubs, business plan competitions, etc. We launch collaborations with other institutions and industry partners to drive new solutions, such as the Research Affiliate Program that Peter was describing at the beginning. And so this Research Affiliate Program works in a way that we actively seek engagement with industry around water and food challenges. Of course, the water and the food and ag sectors are core to what we do, but there are so many other industries that are affected, such as mining, semiconductor, and so we work with those industries too. So for the folks here in the audience that have hard problems that existing products or services can't solve, please connect with me on LinkedIn or shoot me an email. and we can try and explore ways that MIT J-WAFS can help. Can help by leveraging those cross-disciplinary talent at MIT to find breakthrough solutions. And here is a snapshot of our key research areas that we focus on. I'm just going to leave this on the screen for a moment. And then I'm going to invite our MIT speakers to share their latest research on PFAS, a topic that is important for all of us, and also share various ways that you can collaborate with MIT researchers. Well, thank you very much.
Okay, I think hopefully you can all hear me and see my screen properly. A little.
Jim, click that little button in the center, continue to share.
Oh,
You're little, I know that
there
happened
we
before.
are. There we are. Okay, that's a new one.
Beautiful. Thank you, sir.
Okay. Anyway, it's a pleasure to be here, and I've also benefited from the J-WAFS program, which actually really got me going in water and food technologies. And my research group is focused on sensing largely, and I'm going to tell you about detection of PFAS materials using electronic polymers today. This work was funded through J-WAFS through a connection with an industrial partner, Xylem, which is a large water treatment company, and also has had follow-on funding from the NSF through our Convergence Accelerator here at MIT. Now, I think as all of you know, PFAS is a problem, and it has serious health consequences, and the EPA has put forward regulations that will go in effect where people are going to have to test water systems that are particularly drinkable water systems. And the key PFAS members they're worried about are these surfactant types of materials here shown down below. And they all have acidic groups with them that tend to ionize in water. And the EPA would like to be able to detect these at 4 to 10 parts per trillion, or ensure that water levels are below that. Now, the current validated methods are very time consuming. They involve extraction of the PFAS from water, and then liberation and put through a mass spec which is a complicated machine to manage. And so what we've been really interested in is we can create simple point of use methods that can give you that sub-part per trillion levels of detection really out in the field with things that cost very little. And that's kind of the game plan that we have using novel materials and methods. And so what I'm showing here is, a polymer. I'll show you more about this up above. It has perfluoroalkyls attached to it. This polymer is insoluble, so it's not leaching those, but those give it high selectivity for the perfluoro surfactants, or the PFAS materials of interest. And we've also done this electrically. I'll show you that. And the electrical system is actually quite useful because we have invented and have a number of patents around placing chemi-resistive things that change their resistance in response to a chemical, chemi-resistive materials in line with the resonant RF circuits in passive RFID tags, which can be read and powered wirelessly, in principle even by your smartphone if you're looking for threshold detection. Okay, so one of the nice things about our system is we can use it in many different ways. It's a polymer. It can be coated. It can be made in nanoparticles. I'll show you some data from nanoparticles, but we've also, if we coat it onto tubing, we can use it to do continuous monitoring of, for example, industrial waste streams. And the reason we gain such high sensitivity in these materials is that the polymer that we show up here is electronically delocalized. These double bonds here are what we call a pi orbital system, which creates a semiconductive structure where we have a filled band of energy levels called the valence band and an empty band called the conduction band. And when a photon populates an electron from the valence band to the conduction band, the two carriers, the electron and hole, are coupled, and they move together in something that we call a physics quasi-particle, exciton. And if it runs into a low band gap or other places in the polymer that can do electron transfer, but in this case, I'm going to show you if it runs into a place where an analyte, I think, for example, group PFAS binds, it will create a site-selective luminescence from that place. And so what happens here is these excitons essentially migrate throughout the entire system. They migrate through a thin film looking for that one spot that has the PFAS located. And we have found in some previous systems, this exciton can sample as many as 20,000 different sites along the polymer. So that gives us amplification because it's just wandering around looking for an activated site. Now, the way we create activation in these is we have these polymers that again have these pi systems, and you might notice there's this pyridine, this nitrogen here in the ring. And with the perfluoroalkyls tend to essentially sequester into the bulk of the material the PFAS we're looking for, and that PFAS brings a proton preferentially rather than a sodium ion because this is a very hydrophobic environment. And that proton protonates the pyridine ring, and that shifts what we call a charge transfer electronic transition to the red. That creates a red shift in the emission, which gave us that color change that showed you before, and I'll show you again here in a moment. Now, we've done this a number of different ways. One way that works really quite well is to make nanoparticles that we can disperse. This allows for them to have the most efficient diffusion of the PFAS from the water into the particle. We have a number of different polymers that we've done. This is kind of a confusing diagram here to kind of capture all of them, but I'll show you a couple specific polymers here in just a moment. So if we take these and we disperse them, and we can find that very simply with this method, we can get down to 10 ppt. We have actually other methods at a company that we founded that get down below that, but this is what our published methods are. And this is just showing what the data looks like. So we have a polymer here. It's got perfluoroalkyls. This one has these little wings to give it some porosity, which we find to be kind of useful. And what you see is as we add PFAS to this, we get a decrease in one emission band and an increase in the other. So the decrease is in the emission of the polymer that is unperturbed by the PFAS, and the increase is of sites that have been activated by the PFAS that are giving us this green color. And so you can see it highly activated at 10 part per billion, and it's all green. And then you need an instrument in this case to detect these other smaller changes. But an advantage of this ratiometric detection is we can get quantitation out of it too. So we can use these two signals, one going down, one going up, as essentially an internal standard to give us quantitation. Now, we have a number of different polymers we've made that do this. This one actually gives a more, at least to your eye, or in this figure, gives a very clear kind of color change here. You see at at 500 part per trillion, you can actually detect this visually just looking at it in this solution. So we have these nanoparticles in solution. They're being activated by this, and we see detection. And then we can also play games with having something a little bit different than the standard pyridine. We have a pyridine now that has two thiophene on it. That gives us even a bigger shift to the red here. And now you see we get something that's even yellow when it's heavily activated. Now, as I mentioned, we also can do this electrically, and that has some advantages because we can put it into our radio frequency identification tags. But we've also been really interested in thinking about things that maybe could be used by consumers. And we're all familiar with COVID tests that we have used actively in recent years. And so what we have done is created a electrical version of a lateral flow assay, which is what a COVID test is. And in this case, the COVID test, you're picking up antibodies and you're getting, a optical signal. However, if we put what is known as a conducting polymer in place of that optical band, we can make use of the fact that the conducting polymers with small amount of carrier injection into them, that is holes in this case, that we can get many orders of magnitude change in their conductance. In fact, is these polymers often have ranges of conductivity with doping that exceed 10 to the 10%. And so that's really, really a large signal. And so how we go about doing this is we work with a polymer called polyaniline here. Polyaniline shown in the upper right as these radical cations, so it is actually conductive in this form. But if you treat it with base, you take away the protons from this, and you create a material which is an insulator. Okay, so we make an insulating version of this. We disperse it now with a fluorocarbon surfactant, a large high molecular weight fluorocarbon surfactant that is not water soluble and does not leach away. And we can take this as a Krytox grease, for those of you that know your fluorochemistry. And we basically coat it onto a lateral flow membrane. It's a nitrocellulose. And we just wick water up through it. And the water that we wick up here is less than 100 microliters. But it pulls PFAS with it, and it protonates the polymer and essentially converts it back into its conductive phase. And so what we can do with this is we can measure the conductivity as a function of the concentration of PFAS. Here we're doing it for perfluorooctanoic acid. I should have mentioned that was also perfluorooctanoic acid, which is a particularly nasty one, was in all of the other data I showed before. That's what PFOA is. And we show that just with a very small sample size, these are very small little lateral flow assays, we can get 400 part per trillion detection. If we now actually make a filter paper and we can dump, say, for example, a liter of water through that filter paper and then measure the conductivity change on that, we go considerably below subpart per trillion, which actually gets into a problematic range because most every water supply has, some PFAS in it. So we can detect PFAS at ultra trace levels with this. But again, it's also compatible with our wireless method. And we're working constantly on new methods to detect PFAS. I'll show you just one here that we published last year also on that makes use of some impedance spectroscopy to detect PFAS and get at, in fact, is some of the speciation of the different PFAS materials. And like all of us at MIT, I'm fortunate to work with a great group of students. They're very hardworking, and they're the ones that do all the work. I had some of their names on the slides, but we don't really have time to go into it in more detail. But I'm happy to answer any questions.
Thanks very much. I think Longy, what do you want to do? You want to go directly to the next thing? You want to spend a few minutes with Tim's questions? That's a very nice looking dinner, by the way, Tim. Was that xylem?
Yeah, no, I tend to take the group out to a nice dinner.
Lovely.
Most years. Last year I didn't get to because we had some finances, but I make them dress up for this photo. But anyway,
Nice.
I think we do have time for questions because I made it in exactly 12 minutes as long.
Perfect. We got a bunch coming in. Let me start with Robin Newmark. Robin, why don't you, can we unmute Robin and she can ask her question directly? I don't know, Brian, if you can find Robin. Robin asks, she's interested to learn about the current cost and measurement and your estimated cost reduction at commercial scale. So, tell us a little bit about, what are the main cost drivers for making a sensor like this?
Okay, so yeah, so this is let me just maybe go here. So if you look at this tubing will has a less than a microgram per centimeter of polymer in it. And that's actually that's plenty. That's actually more than is necessary. And so the polymer is really not going to be a cost driver at all. It's really superfluous. It'll be more about the other features. I think we have prototypes for instruments that will be very low cost. We plan on providing through a company we've founded called Fluoridics. we're providing that to be kind of a service company where we can provide essentially data analysis and logging to people if they like, or if they want to do it on their own, we let them do that as well.
Cool.
But I think the cost per test will be low. I mean, right now to do a mass spectrometry test is $300 and takes a couple weeks to get it
Yeah,
back.
that's right.
I don't want to say exactly what our costs will be, but they'll be very low, and we'll probably, I think we plan on offering tests for around $20 a pop.
And literally, how many seconds or minutes does it take to resolve the signal in this sort of configuration?
In the protocols we're doing at Fluoridics, which we're doing for some commercial customers, we make the test in 30 seconds.
30 seconds. Excellent. See, time is money, Tim. So there you go.
So a follow on to that. I don't know if you can hear me. Thank you for that.
Hey, Robin.
Follow on to that is a sensitivity in terms of the conditions under which you would deploy these sensors. You know, temperature, et cetera, because a lot of the places are not actually in somebody's kitchen or in a manufacturing discharge station.
Yeah, so we haven't done extensive temperature studies, but we have been, and this is non-MIT stuff done through a company. But when you start trying to sell things to people, what they do is they give you their challenging samples.
Yeah, give you the worst one.
They give you the worst ones, and they give you, and you got a day to give them back numbers, and they know the real numbers, and you give them back numbers, and if you hit within their sweet spot, then they're happy. And we've been able to do that with landfill leachate, as well as a semiconductor waste stream, as well as some things from Air Force bases where there's contaminated water at military bases. So we've been able to hit with 3 complex samples, okay, in a lab. We didn't take it out. It wasn't in the middle of the winter outside or something like that. We do tend to sometimes use, if the pH is way off, we'd have to worry about that. But we also have a standard buffered solution that we use to kind of mitigate those variances.
Tim,
Thank you.
Tim, that begs Paul's question about, you mentioned a little bit complex water matrices. So leachate, that's a yummy one. Are you doing any pre-treatment, et cetera, before you introduce that raw complex water sample into the analysis process?
I believe the leachate that was given to us had been coagulated or filtered somehow.
Okay.
There had been some sort of pre-treatment, and part of it was a pre-treatment of the native leachate, and then something after it had been purified through a foam fractionation process.
Yeah. Okay. Very good. Longy, how are we on time? Do we need to take more questions, or we need to go on?
I think let's move to the next speaker, and then we'll have some time at the end for group questions. Thank you.
All right, hopefully you can see my screen. All right, I see some head nods. All right, so thank you, Tim, for introducing PFAS. So maybe I don't have to repeat some of the concepts, but my name is Aristide, and I'm an assistant professor at MIT. And this is a project that I would like to share on some work we've done on using 2D metal organic frameworks, or MOFs, as potential sensing materials for PFAS. So I wanted to, maybe spend a couple seconds on why we care about PFAS. Maybe I don't have to spend that much time, but I grabbed this map actually from this environmental working group back in 2021, and these were kind of sites confirmed to have PFAS contamination, especially in drinking water. And today I went actually this morning and I looked at any updates I might have, and this is what the map looks like. So with the EPA updating the recommended levels, and also folks sending water samples for testing, the US is showing kind of staggering evidence that we have a lot of PFAS in our drinking water. It seems to be everywhere, despite the fact that we know how harmful this is. And so, yeah, so this website kind of gives you kind of the levels, and also the known sites, about 10,000 sites across the US, and more to come as more testing can be capabilities become available. And also maybe I don't have to tell folks where PFAS tend to come from. We have plenty of sources where PFAS come from cookware to fire extinguishing, even food packaging. So we have plenty of places where PFAS come from. So perhaps we don't have to spend time on that. But since today's topic is about water, I wanted to give you maybe a case that you might not have thought about, how maybe the manufacturing industry is sending PFAS into a waste stream. And I wanted to pick a case on chip manufacturing. As it turns out, when we make our wafers and chips, we are rinsing these things quite a bit. And most folks will say, we have some PFAS during etching and development of circuits, and we can rinse our wafers to make sure, we get high performance chips. But oftentimes these steps release a very small amount of PFAS per step, let's say. But overall, we tend to release a lot of PFAS at the end because the wafers are rinsed from the front end all the way to the back end. So if you haven't thought about it, perhaps if you're interested in or if you work on chip manufacturing, maybe think about whether your steps might be releasing PFAS. And this becomes an issue because usually in a fab, let's say in a clean room, the chemical waste or the we usually have aqueous waste and the chemical waste. So the aqueous waste usually is deemed harmless because the detection methods that we have usually don't show either you have some PFAS. So it could be releasing very tiny amounts of PFAS every day. So I wanted to give this case because perhaps most folks don't think about it. And of course, this is something that has been confirmed using a mass spectroscopy. This is a work out of Cornell confirming that indeed in a lot of clean room facilities, we do have high concentrations of PFAS, those we suspect, even the ones that we don't suspect. So I just wanted to give you this example because I realized in my recent interactions that folks who work on manufacturing don't think about how much PFAS they could be releasing. So now let's go back to our own story. So for my group, we are a materials research group. We're interested in developing materials for a variety of applications, and today I'll be spending some minutes on the detection. But we're also interested in capturing PFAS and also, as of recent, electrochemical defluorination of PFAS. In all these cases, we're interested in these metal organic frameworks. These materials have they tend to have high surface area, high porosity, and they can be made both conducting and porous oftentimes at the same time. So we have some work on all of these three fronts. And as I said, we mostly interested in designing new materials. And this was came about because we think that even though materials such as polymers are promising, but there are other class of materials out there that can be used for protection, for instance. Again, we're interested in these small molecule PFAS or surfactants, as Tim referred to them as, where we tend to have these fluorinated tail and these hydrophilic acidic heads. So these are tricky to detect and capture because they're very small and they're amphiphilic. They can swim through water without interruption. So we're interested in how we can target these both for detection and capture. And we view this as a material science challenge because, again, these are amphiphils, so we need to figure out how to target both ends of the molecules, and in this case, for efficient detection. So this started as a seed grant actually with J-WAFS, where we set out to figure out how to screen existing materials that can be to screen materials that could have a high affinity towards PFAS. And so we've done quite some work on this side where using high throughput screening, focusing on metal organic frameworks, we have identified a few materials that we think offer some specificity and selectivity. And we've also done resistor level iterations. And right now we transitioning to this to my right side here, where having identified a few materials that are promising, can we go into the low cost and scalable manufacturing of these materials into chemi-resistive devices? And here, by chemi-resistive, I mean materials that change their conductivity upon contact of PFAS. And of course, we targeting real-world deployment, especially for environmental cleanup sites where these resistors can work. Our method, again, so here we didn't want to take the approach that Tim is taking on conventional conjugated polymers. Here we're interested in conductive metal organic frameworks or MOFs. where we have thousands of structures of MOFs that we can think about. But here we're interested in the ones that are electrically conductive because we want to be able to read out the response from PFAS contact. And here we also can use the porosity and the tunability of MOFs to get other parameters such as the affinity and also the selectivity. Throughout our screening, we've landed on a few building blocks. So these structures that I'm showing here, maybe I need to get my laser pointer, if that makes it easier. Maybe not. I can use my pointer. But these are the building blocks that we settled on, where these are conjugated ligands that we use to make these frameworks, and we're using coordination with the metals such as nickel and copper. You essentially make a 2D porous polymer, which does not leach into water. And we're interested in how they interact with PFAS. Here we collaborated with Professor Kulik at MIT on evaluating the interaction level with these materials. For example, if you have a metal site in these coordination polymers, could we identify and optimize how these materials interact with different PFAS? So we started with a computation the viewpoint, and as I mentioned, we settled on a few candidates here. And again, once we identify which ones have strongest affinity, we're interested in these resistive devices where we're measuring the change in the current when these materials bind with the PFAS. And we can do either just a regular resistive device, or we can do spectroscopy such as impedance spectroscopy, as Tim mentioned a little bit. The spectroscopy offers higher sensitivity, as I'll show in just a second. And I think that's where we're having more success. So the first thing we wanted to do was actually show that we can make these materials into thin films, because this is tends to be challenging. For TIM system, for the polymer, you can make a solution and you can cast it on substrate. But for these metal organic frameworks, usually you synthesize them as a powder. So for us, we had to investigate how do we grow these materials into thin films onto electrodes. So we can do this solvothermal method, where we do the metal and ligand kind of sequential dipping of a substrate, and we can grow these films actually quite high quality, and it's very robust. We can even use a robot to do this kind of dipping, so we can make thin films that are down to 100 nanometer thin onto a variety of substrates. We can do ITO, we can do silicon, we can do glass. So we believe that with in this case, we're using copper as our metal node, we can make these coordination polymers in a low cost manner. And of course, now we're interested in how they respond. And I don't know if you can see it on my right side, but we can attach, or you can even do so this is a picture of a rudimentary version of our device, where you can just attach two cables onto working ITO electrodes, and we can dip it in a water sample. And of course here, this is a question that was mentioned earlier. We use a buffer, a PBS medium for detection in this case. So after figuring out how we can grow these materials into thin film, we're investigating the resistor response. So here I picked a couple graphs here. Maybe the one on the left shows from the initial conductivity of the MOF. So the MOF gets doped by the presence of the PFAS. Here we're still figuring out how selective this is, or how it can distinguish between different PFAS. But as you can see, the current gets higher, almost more than double as you increase the concentration of PFAS, in this case in a buffer solution. I think the graph on my left, on my right, is actually extracted from these now impedance spectroscopy measurements, because these are more sensitive as we go to much lower concentrations. So here we're using something like a biologic electrochemical setup to measure the change in the resistance of the material, where the metal organic framework is a working electrode. And you can see that as we increase the concentration of the PFAS, here in this case, PFOA and PFOS, we're seeing different responses at different concentration. And so this is something that we're also interested in, where these materials, how many times can we use the same working electrode? It turns out it actually works quite well, especially if you have very low concentration of the PFAS. We've gone down up to 10 times of repeatable dipping and rinsing in water. Even the structure of the metal organic framework here remains unchanged. Something that is exciting, especially if we go into putting it in, let's say, in a continuous flow setting. So again, as I mentioned, we are a material discovery group. We spent quite some time on this side here on high throughput screening and a large area processing. We have made this kind of test kits that are chemical resistors, but we're interested in also in using them as working electrodes in impedance spectroscopy measurements. And again, I'll save it for our discussion for some questions. But how do we transition to more complex water matrices? For example, muddy samples or clean room samples that have other contaminants. That's our ongoing effort. And let me stop here so that we can answer some questions.
All right.
All right, I flew through those, but I believe.
So I'll start with one. So in general, I mean, the PFAS family of compounds is very wide and very diverse.
Yeah.
And I was thinking about all of our discussions so far about developing sensors.
Yeah.
And are we, because a sort of a portfolio of PFAS might be present in any particular water sample, do you have to be careful about discriminating one sort of type of PFAS relative to others? Do sensors glom onto a variety of PFASs, and then you'll have to make some assumptions about what the aggregate PFAS load is if you just grab a couple sample molecules? So tell us a little bit about how you do a sensor measurement when the thing you're sensing has got thousands of varieties.
Yeah. So I started. Let me start with this slide. Try to answer that one. So here I showed this, different chemical size and molecular size of these PFAS, and of course also a variety of functional groups that we can think of. I think this is why we wanted to choose these metal organic frameworks. So we think that with some kind of size exclusion, we should be able to get some discrimination, because we can actually engineer the porosity. So Tim alluded a little bit on the porosity in his polymer, where they make this kind of star-shaped monomers in the polymer. For us, we get that for free in the frameworks, because the framework with the ligand design, we can choose how porous the material is. So through the size exclusion, we think that we can discriminate between the long and the short. But when it gets ultra short, by ultra short I mean let's say right when it's down to like 4 carbons, that part has been challenging so far. So we've been successful with 10 carbons to 6 carbons. Those ones we believe we can distinguish at low concentrations. When they get much shorter, it becomes challenging. You're spot on.
Yeah. Okay, good. Thank you.
Yeah.
All right, Longy, you want to keep going? All right, let's do it. Thanks a lot, Aristide.
Yep.
Present. So my name is Benedetto Morelli. Thank you for the opportunity to present. I'm an associate professor in civil environmental engineering, and I'm the mission director for the MIT Climate Project when it comes to the wildcards mission. Today, we're going to talk about biomaterials that can be designed for water filtration, and in particular, we're going to see membranes that are based on natural silk and cellulose to remove PFAS. The good thing of talking last is that I don't have to introduce PFAS, so I just have to talk about the technology that I developed. But I just want to echo what Aristide presented of the whole spectrum of activities that we can do around PFAS, but that comes to detection, captures, and abatement. And of course, from the title, you can guess that this technology is specifically geared towards the capture of the PFAS. And so the idea is that you have water flowing through a membrane, and the membrane is able to separate the PFAS from the stream. So what are the biomaterials that we're talking about? Biomaterials are typically conceived as materials that can work inside the body to provide the function, but in reality, we can expand the notion to also natural materials and biopolymers. So these are materials that we can extract from commodities, from biomass. And during the extraction, we're able to maintain the structure of the materials, but then we can also add functionalities when it comes to provide new formats to the material. So we're able to take the materials that can come from, as we said, natural sources or synthetically engineered. We nano-manufacture them. Typically, this happens at room temperature, atmospheric pressure in aqueous environment. We can add dopants, and at the same time, we can exploit the self-assembly of these materials to make engineered forms and function. The merits of these biopolymers is that they are edible, typically non-toxic, and they biodegrade. We can choose how quickly they can biodegrade. The challenges, particularly for their application in food and water, is the scalability, how easy they can be manufactured to have impact, and being able to retrofit existing techniques and also the needs of the stakeholders in this field. So the materials that my lab typically use is silk. We don't start from the silk that you consider a high expensive textile silk. We use the silk that comes from the waste of silk. The fiber itself can be deconstructed by removing the hydrogen bonds that keep the silk fibrin protein together. This can be done using a chautropic agent that is able to disaggregate the hydrogen bond of the beta sheet in silk. And the fiber itself goes back to what it is, as you can see here, a water suspension of silk. And then by driving the self-assembly again, silk from a fiber can become a technical materials that can compete with synthetic materials to perform in a wide breadth of applications. So all the materials formats that you see here are done with molecules of silk that has been self-assembled to impart a new form factor. The same principles can be applied to other materials that we can extract, both proteins and polysaccharides. So we're thinking about, for example, cellulose, we think about chitosan, we think about, in some case, keratin or alginate. In the case of silk, my lab has shown that it can be scaled up to have impact. So this is just to give an idea that these materials are real and can be scaled at the right cost to have impact on large commodities. In the case of silk, we scale up the application as a food coating to extend the shelf life. And now several thousands of tons of leafy greens are treated every month in the United States to extend the shelf life, and they're available as a product on the market. The company is called NAWI, and the scale-up process has been done in collaboration with Canon, Virginia. So this is just to tell you that the streamline of these biopolymers to function and to impact is alive. And so what my lab developed is basically the application of this material to make membranes that can be used to extract PFAS from water. Particularly, we started from cellulose nanocrystal that we extracted from the pulp of wood. The cellulose nanocrystal in solution can template the growth of silk. Typically, silk, if it's self-assembled by water evaporation, forms monolithic material. In this case, the CNC templates the growth of silk in nanofibrillar matrices that are highly ordered. The big advantage of using the CNC is that the CNC can be functionalized in a positively charged nano whiskers that can be designed to be able to electrostatically interact with PFAS, as Aristide told us before. Electrostatic and hydrophobic interactions are key for polymers to interact and also other material to interact with PFAS. So in CNC, we can bring positively charged, so we can design positively charged in CNC to help with that, but also to impart anti-fouling properties to the membrane. And the silk itself assembles around the CNC in very hydrophobic structures called beta sheet, and this beta sheet, of course, also contribute to their interaction with PFAS. So to make a long story short, we can grow these nanofibrils in water, then eventually we can use vacuum filtration, we can compact them, and we end up having membrane of different thickness. We can go down to hundreds of nanometers to several microns. These membranes are extremely robust mechanically because of silk and nanocellulose are robust polymers. They're bendable, they are flexible, and they can be used to filter water by applying pressure to them. They have a nanoporosity that can be engineered by changing the parameters, the knob that regulate the self-assembly, so the size of the nanofibrils and the growth rate of the nanofibrils in water. They can be used, this membrane, to remove heavy metals. For example, in this case, we show that we can remove nickel, cadmium, chromium, lead, and a little bit of thallium. And this is the performance that you get when it comes to heavy metal removal, which goes to very high level. But at the same time, oops, okay, did I skip anything? No, at the same time, they can also be used to treat PFAS. In this case, just to give you an idea of what type of solution, this is just PFAS in water. We're not at the point, or in this data particularly, we didn't show yet complex solution or complex fluid where we have other contaminants present. It's just the PFAS that you see in the schematics. So typically we target long and short PFAS. We show the differences between a negatively charged cellulose nanocrystal compared to a positively charged, and so the charge of the nanocrystals provided a big impact in the filtration capacity, as you can see in the sorption isotherm, which were conducted in the first 8,000 bed volume. We know that this material is able to resist to high variety of pH. Particularly, we can go from anything that is between 1 to 10 in terms of pH, and typically the performance of the PFAS absorption is not influenced by variation of pH in this range. Being these biopolymers, if we go to pH that are either too high or too low, we end up destroying the biopolymers, and so that would be something to consider. When it comes to temperature stability, these biopolymers are stable in a range that goes from basically the freezing temperature of water up to almost boiling of water. The materials itself is stable up to 180 Celsius, but this is where we tested it. And when it comes to the performance of our membrane, we see that we have a very good performance when it comes to the filtration when we compare it with the sorption capacity, where we compare it to existing technologies that have already been developed for PFAS removal in the forms of a membrane. To conclude, I would say that this is a technology that we're still developing. We started with silk and cellulose nanocrystals because those were two materials that we had available. In the lab, we're now expanding the portfolio of biopolymers that we're using to generate these membranes, and we're testing how different biopolymers that are extracted from different biomass have an impact on the sorption capacity, the stability of the membrane, the anti-fouling properties, and at the same time of the price cost that we can think we can have. Given that this is, of course, is one of the most challenging part when it comes to PFAS filtration, so be sure that the price is low enough. Another challenge that you have with the biopolymers is the batch-to-batch variation. Typically with silk and nanocellulose, we have very, very good performance, like you have extremely good repeatability. If I have one student that takes over the project from another student and different sources of silk from different parts of the world, we have very consistent performance. For other biopolymers, the source of the polymer historically has always been a challenge. And so the extraction of the biomass has a big role on that, and we are developing protocols that are able to minimize the batch-to-batch variation. The other challenge that we have is that the self-assembling process that you saw at the moment takes time. So it can take several hours. And so we recently achieved a method where we have been able to really speed up the process of the self-assembly down to 2 seconds, if not, single digits minutes. So we're working on the scalability of the technology to try to have an impact. And with this, I want to thank J-WAFS for their continued support to the research on water and food. At MIT, they really provide us with the initial spark that has many of these projects going. And then for the specific project, I have to also thank the NSF and the Office of Naval Research. Thank you. Thank you very much.
Great. Thank you. And thank everybody. This was a really nice, Longy, you pulled together a great portfolio of leading research in this area of developing new sensors and measurement technology. So you all, thank you very much. We have a bunch of questions, and actually one came through the Q&A section from Kyle Schoenheit. And so, Ryan, can you find Kyle and see if you can unmute him? Or Kyle, maybe you can unmute yourself. I'm not sure. Let's give that a try.
Peter, can you hear me?
Yeah, you're a little quiet, so speak up.
All right. All right.
There you
Thanks, Peter. Thanks for the invite, and thanks for the presentation so far. I truly appreciate it. You know, a couple of questions around the commercialization of the opportunities that you share, and one is actually a jump off for your question earlier, Peter, is the identification of what type of chain of PFAS, right? Because it's going to help determine the treatment required, right? So whether it's short chain or long chain, if you can give some flavor around that, if you can identify the differences of that. And the other one is we've been looking for a sensor. Xylem has been looking for a sensor to identify PFAS probably for several different years, right? So identify it, treat it, and then discard it or destroy it, right? Is our three legs of the stool there. And one of the pushbacks is the accredited laboratory acceptance of the sensor versus using the sensor more of an operational reflection audit of the treatment process. If you can just add some color around that also.
You guys can make a comment. I want to make a comment too. What do you guys think about accreditation or, kind of qualification of a new sensor relative to established methods? Don't all shout at once.
Yeah, I mean, the certification, you're thinking EPA certification process. You know, that is definitely a challenge. That is not something we're pushing for right away in our commercial efforts. Although I do have a new project that I didn't talk about starting with J-WAFS that we hope to be able to get at things that I think will have the ability to detect, to identify the very specific PFAS elements, which I think like a mass spectrometer, but not a mass spectrometer, that could be eventually eligible for certification, but it's very early stages right now.
Okay.
Thank you.
Benedetto or Aristide, do you want to make any other comments?
Yeah, so I think the comment I would make is if you look at existing methods like mass spectroscopy or spectrometry, you have a long procedures for sample prep, right? So I think we could cut ourselves some slack in terms of sample prep. I don't think it's reasonable to ask Tim to go put his sensor directly into the charge right now. I think it's okay. I think it should be there should be some leeway in terms of I can still prep the sample. If it takes 15 minutes, I don't see any problem with that. If it's putting it in a buffer. So for me, I don't see a huge problem in that regard, as long as we're cutting down the time and the cost. That would be my comment on that.
Yeah, Kyle, my thought is why bother getting it certified? I mean, to some extent, sometimes a non-certified method, if it's accurate, can be nice to have because in some regulatory frameworks, certified methods have to be reported depending on who the entity is doing the measurement. And so sometimes if you want to check on something, you don't necessarily want to be obligated to report the number. We see the same issue with DBPs and trihalomethanes that, there are several sensor systems out there for trihalomethanes that are really nice to use, but the numbers aren't things recognized so that you don't have an obligation of your public drinking water system, for example, to report the number. So sometimes non-certified methods have a nice role to play in information as opposed to reporting.
Thanks, Peter.
Yeah.
Appreciate it.
Yeah. I see we had some MOF nerd chat on the chat about functionalizing MOFs, et cetera, et cetera. Again, I mean, maybe you guys, all three of you can reflect on this. The PFAS family of compounds is so wide and diverse that it's an interesting kind of sensing challenge. And I guess, do you have any thoughts in general that when you are going to go after a measurement, Are you going to target one specific subset of PFAS compounds because they're easy to measure and then make an assumption about what the total bulk PFAS kind of load might be? And if so, is it easy to correlate a subset of concentrations that you can measure accurately with the bulk?
Mm, mm. Tim, you wanna give it a try first?
I mean, I think you're right. There can be a depending on what samples you're working with, there can be many different types. I mean, part of it is just a total PFAS. I mean, in some sense, I look at our stuff as like a warning, like boom, you got something that's above a certain threshold in there,
Yeah.
and in that case, you need to figure out what it is. One of the things that is kind of interesting that's come out is that if you look at really diverse PFAS families, sometimes if they're like 40, 50 compounds, mass spec can let you down because mass spec sees
It
like
spreads it out, right? You got all these little
for
masses, so it's
everyone.
just a schmoozer. Yeah.
When in fact is people have found that high field NMR
Mm.
can do total fluorine
Yeah.
much more effective and in fact is like the project I have funded that we're just starting actually this month with J-WAFS is a new method that will use NMR to try and supplant mass spec.
Mm.
And but the NMR has been shown to be more accurate than mass spec. So with so the EPA methods even break down with really diverse populations.
That's a very, very good point, Tim. Thank you very much. That's great. We have time for a few more questions. Longi, you wanna take us on out with any final comments or questions that you have?
I'm good. I'm actually just going to copy and paste one of the questions that came through and Aristide's response so that everyone can see that question.
Yeah.
Oh, it seems like I might have just pasted into the panelist. I can tell Ethan what my response was to make life easier. So I think his question was whether we see a difference between different functional groups on a PFAS depending on the material that you have. So at least in our experience, it seems like our MOFs respond better, at least strongly towards the sulfonates. Tim, I don't know if that's the same thing you see in your own world, but for the MOFs, we have some design space we can control how electron-rich the metal organic framework is by changing the combination. But I think that answer, Ethan, I think we'll have to do more iterations. We tested on a longer PFAS. Does that translate to ultra short PFAS? I don't know if I can answer that right now. But very good question in terms of the functional groups.
Very good. I see we have a hand raised from Vineeth Ram. Vineeth, can we unmute you and you can ask your question directly? We got to hunt down the control panel and find his mute button. Vineeth, I think you can actually unmute yourself. Give it a try and you can ask your question. Oh, we're trying. Oh, Vineeth, we still see that you are muted. We want to know what your question is. If we can't find you now, we will definitely find you later. So I'm sorry about that. Anyway, everybody, on behalf of all of us at NAWI, one of the things that's very valuable for us is that, the water treatment, water measurement community it's important for us to stick together. So this is why we love having these periodic sessions with J-WAFS. And Longen and Tim, Benedetto, Aristide, thank you again so much for spending time with us today. And as we will go out, I'll simply say that this will be captured in a recording and available on the NAWI YouTube site. And you can also follow up with the individuals. And don't forget that J-WAFS has a very active affiliates program, and you can get more information on their website there. So from all of us at NAWI and all of us at MIT J-WAFS, thank you all again for coming out today, and thank you for this fascinating set of talks. And we look forward to being able to really push forward in understanding this broad class of problems with perfluorinated compounds. So everybody, thank you again so much, and have a great rest of your day. Bye-bye.
Thank you. Bye.